So I'm recording now. I'm going to share my screen. Not what I wanted. And stop share. Try it one more time. Wonder if it's. So I think you're all seeing one big PowerPoint slide. Is that true?
Yeah.
Yes.
All right. So this is where we left off with hydrogenotrophy. I still cannot say that word. And this is the one excellent example of the chemolithotrophy type of metabolism where they're using these inorganic nutrients to strip electrons off of. We can't do that. We always have to use an organic molecule like glucose when we start working with that and running it through a reaction. So they're very flexible in their physiology in this regard. They have, depending on the microbe, there's a lot of different molecules that can be used for this. And so hydrogenotrophy is a big one. Hydrogen gas is, chemically speaking, very capable of having electrons stripped off of them and then getting fed into different reaction pathways. This is just one example I'm showing you. There are other options out there, depending on the microbe of how they work with hydrogen gas. But the big picture idea is that this hydrogenase enzyme is capable of grabbing electrons, so to speak, and stripping them off of hydrogen. And in this case, running it through a real small electron transport chain process. And then, where's my laser pointer? And getting this electron flow to occur, and then coupling that to getting a proton gradient established. So in key steps, you see protons getting pumped across the membrane. And then here you can see there's a buildup of hydrogen ions on one side of the membrane compared to the other. So you get this proton gradient allowing for the diffusion of protons through the ATPase to generate your ATP. And then most chemolithotropes can take in CO2 and use that to build their organic matter that they need because they're organic organisms. So they need organic molecules to build and repair and reproduce and so on. So really kind of an interesting lifestyle, and again, it's what habitat they evolved in, where they, through natural selection and selection pressure from the environment, these enzymes, these special enzymes and pathways evolved to capitalize on all, in this case, all the hydrogen gas available in their environment. Well, another interesting idea in the world of microbes is photosynthesis. And it's not obviously just microbes that can do this. When I teach plant physiology, we get into this in pretty good detail. And so certainly plants, and that's probably what you are most familiar with. I'm guessing anytime you've been taught photosynthesis, they've probably focused on plants, not cyanobacteria. And probably, I would bet most of you don't even know that there's other ways to do photosynthesis when you get into the microbial world, unless you've had a micro class already. So here, this is how plants do it. All right, so here's a chloroplast that, they say, plants have chloroplasts in their leaf tissue primarily. And inside the chloroplast are these special membranes. So here they're just showing a little bit of the membrane that's inside the chloroplast. And that's where all the chlorophyll pigment is embedded. So chlorophyll is the workhorse pigment. Chlorophyll is absorbing that light energy. And during that process of absorbing, that chlorophyll pigment is absorbing light energy, it actually excites the electrons in the chlorophyll pigment, and it kicks off this electron transport chain process. Here they're not really showing that, so I'll get into the electron transport chain in a second here. This slide is just showing the kind of the big picture idea of, in total, what's going on when you think about photosynthesis in the plant. And so they're linking both what we call the light reactions, where the chlorophyll pigment absorbs light energy, and through electron transport chain process, the end result is being shown here. ATP gets produced, because you get a proton gradient during this whole thing as well. And so you get some nice ATP, and then you get another very important molecule called NADPH, which is similar to NADH. And it's what we call an electron carrier. So this molecule is very happy, chemically speaking, to now donate electrons into some other pathway to get that cooking along. And that's what they're trying to show here, that once the light reactions do their job and you get ATP and you get NADPH, then it feeds into this pathway that we call the Calvin cycle. And it's a cycle, obviously, it's going round and round and round, but that's where you see, I call it step 2, phase 2 of photosynthesis. And it's the biochemical reactions where you finally see what happens to the carbon dioxide that the plant brings in to its body, into its leaf tissue, and then feeds that into the Calvin cycle, and you end up getting some sugars produced through that. So in total, when you look at what's going on in total, you get ATP and NADPH being produced, and then CO2 gets, what we call it, gets fixed. They often use fixation. They say CO2 is fixed, or carbon dioxide fixation. What they mean by that is CO2 is brought into the plant and sugars are made. All right, so this is like the big picture idea of photosynthesis in the plant world. When you look at the different, so here in, when we look at leaves, they're green mostly because there's so much chlorophyll pigment, and the chlorophyll is what's doing all the absorption of the sunlight energy. Well, there's other pigments though too that you don't see with our eye because there's so much chlorophyll. Well, now in the fall, we start to see some of these other pigments. However, with the beautiful fall leaves, the color change, the chlorophyll, what's happening in general is the chlorophyll is getting broken down and some of the other pigments are revealed, and you can see them with the naked eye, and it's just beautiful in the fall to go hiking in the woods. I hope you do that again this weekend and check out to see if there's any fall color change going on. But here what we're looking at is an absorption spectra where they isolated different pigments from photosynthetic, mainly a lot of this is from plants, but some of it's from bacteria too. And when you run it through a spectrophotometer and take an absorption spectrum, you can see that different pigments will prefer, if you want to think of it, they prefer certain wavelengths of light. And what I mean by that is you can see these peaks in absorption. So this one, I think 3, let me just double check what 3 is. Oh, I can't easily switch. 3 is chlorophyll B, 2 is chlorophyll A. Let me go back, make sure you're seeing what you're supposed to see. So 2 is chlorophyll A. Yeah, that makes sense. Are you seeing the absorption spectrum slide again?
Yes.
Okay, thank you. All right, so the absorption spectrum, so 2 is chlorophyll A, so that's the real workhorse pigment in the plant. And you can see that it prefers, so to speak, because of the peak in absorption, it prefers to absorb blue light and red light. And then other pigments, like 1, I think is from a bacteria. 1, you can see it prefers way over, beyond the visible spectrum, what our eyes can't even pick up. And then over here, way over in infrared. So, and I think 3 or 5, carotene is on here somewhere. I don't have it all memorized, but you can see what I'm trying to get across here is that different pigments will absorb different quality or colors of light. And so not all pigments are the same. When they're getting hit with sunlight energy, it's not, they're not all trying to grab the same quality of light or the same color of light. And that's really kind of nice, actually, because if you think about complex ecosystems where there might be a variety of photosynthetic organisms, if they were all vying for the exact same quality of light, that would be really tough. There'd be a lot of out competition, and then only one type of organism would dominate, and you wouldn't get a very complex, diverse community. And so the fact that you can have many photosynthetic organisms all hanging out, even shoulder to shoulder, so to speak, or one on top of the other in a water column, for example, they can be layered in the lake or stream, and they can all be doing photosynthesis because they can absorb different qualities of light compared to each other. And so they can all kind of like get along together in the sense of, you know, I like this quality of light, you like that quality of light, we're good, we can hang out together and we're not, you know, vying for the same thing. And so it's really interesting when you start, when you look at a graph like this and you can see that, you know, these pigments evolved to capture different wavelengths of light. And so we will hopefully get to this idea when we set up our Winogradsky column next week and let it start incubating. And one of the things I hope we'll see over time, and actually we kept back one of the Winogradsky columns that's been going for about a year. And in that, I think we'll nicely see this idea that's being shown in this picture of a water column in a lake somewhere, and the different layering that can occur by the different photosynthetic organisms, because when sunlight strikes a water system, some qualities of light, you know, shine only to the surface or very shallow, and other qualities of light can go deeper. And so it just depends on the frequency or the wavelength of the color of light. And so you can see that they're layering here, and the cyanobacteria and algae are on top, and the green, so-called green photosynthetic organisms are next, and then the purple. And so they're all doing photosynthesis, but they're all coexisting quite nicely and getting all their needs met because they are grabbing different qualities of light. So it's fun to think about this when you're looking at lakes and rivers, and, you know, understanding that there can be this layering, and they're all capable of doing their own version of photosynthesis with their own special pigments. Okay, so this is a slide showing just the light reactions. Well, it's focusing in on the light reactions. So most of what you're seeing here is what we call the light reactions of photosynthesis, of how plants do it. Now, I'm not going to ask you to memorize this from start to finish. In plant physiology, we will, but not in this class. In this class, what I would like you to do is, first I'm going to talk about how plants do it, because I think most of you have had a little bit of that at some point. You know at least the vague basics of it. And so I'm trying to use that as foundational knowledge so that then I can start comparing some of the new stuff you're going to learn back to this to show you similarities and differences. So how I talk about this to you, and you'll see me talking about, well, what's similar, what's different? That's how I want you to start studying this material, looking at, you know, side-by-side comparisons and picking out, you know, what are some of the key similarities and differences. That's really what I want you to get in this class, because we're going to be looking at several different versions, whereas in plant phys we only focus on this. This is all they get, and they don't compare it to anything else. So, okay, what do we have going on here? So this is another electron transport chain process that they're showing. So you can see these proteins and pigments are embedded into the membrane inside the chloroplast. All right, so you have to think about the chloroplast, the membrane. There's an internal membrane called the thylakoid, and that's where the chlorophyll is pasted into the membrane, as well as all these other proteins and enzymes. So you see the sun light energy they're showing. Let me get my laser pointer up and running. So you see the sunlight energy is striking these two very, very special complexes. So they're called photosystem 2 and photosystem 1. That I do want you to know, because we need to talk about those when we compare to the other things you're going to be learning. So when plants do the light reactions of photosynthesis, they have two photosystems. So that's important. They're named photosystem 2 and photosystem 1. They both have to absorb light energy in order to keep the electron transport chain process going. We typically start by thinking what's happening what's happening with photosystem 2. So in this photosystem, there's a bunch of chlorophyll pigments, and there's other proteins and other things as well. So the light strikes the chlorophyll in photosystem 2, and it excites the chlorophyll electron so much that it can boost that electron off of chlorophyll and give it to a donor that's waiting to take that electron. And so the arrows here, the black arrows, are showing the electron transport chain process from photosystem 2 all the way over to this last, the NADP reductase. All right, so a couple of things also with photosystem 2. Photosystem 2 is where we see water getting, I often say split. What I mean by that is electrons are getting stripped off of water to replace the electron that got excited in chlorophyll. So when light strikes the chlorophyll pigment, it excites the electron in the chlorophyll and that gets given off. Well, now chlorophyll is missing an electron. And so the chlorophyll pigment will grab an electron from water. Why is that important? It's important because if you strip enough electrons off of water, you end up with oxygen as one of the products. Now, as far as photosynthesis, plants don't really do much with oxygen through the photosynthetic process. The oxygen gets made and then that's it. We don't really consider it beyond that. But it's oxygen, right? I mean, obviously that had a huge impact on what happened in early Earth as the plants were evolving on, especially on land. And so oxygen being produced is a big deal. And so we call this oxygenic photosynthesis. Oxygenic, oxygen producing, genesis means genic, genesis, to create. And so this is an excellent example of oxygenic photosynthesis because electrons are getting stripped off of water. And if you do that enough, you get oxygen. So then you get this electron flow going. As long as light energy is striking both photosystems, it will keep that electron flow going all the way to the end. And here you see NADP plus reductase is kind of like the last step where you end up giving, that will give an electron to NADP plus. And if you do that, you'll end up reducing it to NADPH. So that's one of the huge payoffs when you look at, when you look at this whole thing and go, well, what's the big deal here? Why is a plant bothering to do this? One of the things it's doing is creating NADPH. Super useful for the next part of photosynthesis. All right, so that through the electron transport chain process, you end up getting NADPH. But that is not the only thing. You also, again, we have an electron transport chain process. So right away you should go, oh, I bet there's a proton gradient in here somewhere. And that's true. And so here you can see key steps of the pathway. There's a proton here getting pumped across the membrane. Protons here from the splitting of water also build up on this side of the membrane. And so you get this imbalance again of protons across the membrane. So you get a proton gradient. So that should sound familiar by now. So now you have them diffusing back through the ATPase, and that allows for the generation of ATP. So by the time the light reactions are done, you get two really, really important molecules, ATP and NADPH. Questions on that part of photosynthesis?
So pretty much as an end result, we have to focus on, okay, so ATP is produced through the ATPase and NADPH is produced through the NADP plus reductase.
Yes, yep.
Okay.
So understanding that it's an electron transport chain process linked to a proton gradient getting established, yep, and then you get your ATP, and through the electron transport chain, electron will, so we call NADP plus the final electron acceptor. And so it will grab the electron in the end and then get reduced to NADPH. And so both ATP and NADPH can then get used in the Kelvin cycle. So the light reactions kind of set the stage. They're like, okay, we need ATP, we need NADPH so we can take in CO2 and make our sugars. And so by then, once they get those molecules, you can see it's still happening in the chloroplast, right? Right in the chloroplast, you have the Kelvin cycle, which is where you see the CO2 coming in. And it's a series of reactions. That's why they're showing multiple arrows here, zooming around in this circular path. And so you have several reactions going on there that will allow for the CO2 to get fixed into sugars. And at key steps in that cycle, you have to use ATP to accomplish some of those steps. and you have to have NADPH donating some electrons into the Calvin cycle at key steps. And so again, I'm not going to show you the Calvin cycle in detail, and we're not going to get into that. You just have to know that the Calvin cycle is used by plants to take CO2 and make sugars from it. And you need to have ATP involved in that process as well as NADPH. Otherwise, the Calvin cycle just stops. It grinds to a halt and won't work. Okay, so big picture ideas here, some key concepts I want you to know, but kind of, thinking big picture here. In plant phys, on the other hand, we go into detail, but we only focus on that one.
Quick question.
Yes.
So do organisms that photosynthesize or similar pathways, do they usually have a lot more NADPH than organisms who don't?
Yeah, I don't know that for sure, but that would make sense, right? Because if they're constantly capturing light energy and running the electron transfer chain process, they should be able to make a pretty good dose of NADPH. They probably use it up pretty quickly though too, because they probably crank up their whatever version of the Calvin cycle they're using. Not every photosynthetic organism uses the Calvin cycle, but they probably take advantage of that, high amount of NADPH, and that allows them to make lots of sugars, which is obviously good for them. So, but yeah, capturing it, wouldn't it be great to just go to the beach and lay out in the sun and get your sugars? It'd be awesome. Wouldn't have to cook. Not that I hate cooking, but it just takes time, and sometimes I don't have time. All right, so here, I know this looks complicated, and again, don't panic. I am not asking you to memorize step by step. This is taken from a microbiology textbook, and it's showing how cyanobacteria do photosynthesis, and it's actually really similar to what plants do. So there really isn't much difference. It's just that the artist put in more detail here than what you just saw in the previous slide. But again, if you just sit there and look at this for a few minutes, you go, okay, yeah, it looks like we're looking at a membrane with a bunch of proteins and complexes embedded in it, arrows going, down the line. Yep, this looks like an electron transport chain process again. All right, so it's basically what we just looked at. It's just a little bit more detailed. So you see photosystem 2 on the far left here. You see photosystem 1 in this electron transport chain process, and the ATP being produced with the ATPase. This figure on the top, I get into this in plant phys, but this is one way of, they're trying to demonstrate, they put it on like an energy gradient here on the left. And so when photosystem 2, when the chlorophyll pigments absorb light energy, they're trying to show that that electron gets excited to a higher energy state. So when you go up the y-axis, that's higher energy. And so they're just trying to show visually that when that chlorophyll absorbs light, it excites the electron, boosts it. I tend to say it boosts it to a higher energy state. And then that is like potential energy now that allows for the electron transport chain process to kick in and start zooming from donor to acceptor, all the way down until you get to the final or terminal electron acceptor. All right. So this is basically saying the same thing as what we just were looking at with a little bit more detail. And they're focusing in on cyanobacteria because it was a microbiology textbook. So, but basically you can pretty much say that cyanobacteria and plants do oxygenic photosynthesis pretty much the same way. I don't really want you to focus in on anything other than that, really. Now, this slide, however, is a summary showing that there's more to photosynthesis when you get down into the microbial world. And in fact, when you look at this, you see the oxygenic row is at the bottom here with cyanobacteria. But then all these other examples, they're saying it's anoxygenic. So an on front of a word means without. So they're saying this is without the creation of oxygen. So if it doesn't create oxygen, what were we just looking at with plants and cyanobacteria? How does oxygen get created again when we look at how they do photosynthesis? What happens to get that oxygen?
Water splitting.
Yeah, water splitting. So basically what that's telling you is that, okay, they must not be splitting water when they do anoxygenic, they can't be splitting water, because if you split water, you get oxygen, but this is saying it's anoxygenic, so it can't be water. It has to be some other molecule that they're ultimately stripping electrons from to feed into, to keep the electron transport chain process going. And so depending on the microbes and the type of photosynthesis, you can see different, I'll explain what none means. Well, actually, I take issue with this top row. When I teach Gen Micro, I say this really isn't photosynthesis, because when you look at the strict definition of photosynthesis, this one doesn't cut it. But the authors of this, they grouped it in there, and I understand why they did it, but it's not really photosynthesis. So anyways, but then you see other electron donors. So you don't see water here anywhere. There's no water being used as the ultimate electron donor, of course, except for the cyanobacteria, because they are creating oxygen in the process. So anoxygenic means, okay, we got something else. So you can have an organic molecule that they strip electrons off of and feed it into the electron transport chain. Inorganics, just depends on the microbe, what ecosystem did they evolve to live in. And again, looking at carbon sources, and you can see there's some variation there too. It's not always CO2. Some microbes are incapable. They didn't quite evolve that capability of taking in CO2. And so they kind of get shortchanged on that. Too bad. They never quite made it to grabbing CO2. So again, and then we give them weird names. I don't know why they do this, but you know, the green sulfur, purple bacteria, that's actually how we talk about them. It's kind of weird. But let's look at a couple of examples. I'm not going to go through all of this. This table is just to really get you to appreciate that there's plenty of other microbes doing photosynthesis way differently than what you've learned by how plants do it. Okay, so let's look at the green bacteria. So we often call them green sulfur bacteria because they typically use some kind of sulfur molecule that they'll strip electrons off of. So photosystem 1 is the only photosystem that green sulfur bacteria use. All right, so they just use one photosystem. Plants and cyanobacteria have 2 photosystems. So that's a huge comparison to think about. That's important. That has a profound implication on how they run their photosynthetic process. And so here you can see hydrogen sulfide gas right here is kind of the kickoff point. So they're saying that you have these pigment complexes. Green sulfur bacteria have these really special pigments called chlorosomes. It's actually kind of like almost like an organelle, really, with a ton of these pigments, the bacteria chlorophyll pigments embedded in it. And green bacteria are really good at hanging out in environmental areas that are pretty dim, low light intensity, because they have this chlorosome organelle, although we hesitate to say organelle, but we kind of say structure. But typically, I mean, when you look at it, it's like an organelle, so it's like, get over it, it's an organelle. But anyways, I digress. If you have this chlorosome structure, it allows you to capture this really low quality dim light. It's so efficient that they can still do photosynthesis even in murky water, for example. And so what's happening here, they're trying to show that the light is a lot of it's getting captured by the chlorosome pigments, and then that gets funneled to the photosystem 1 reaction center to really boost and excite the pigment here. Whoops. And then you can see there's this small electron transport chain process going on as you follow the arrows here. And you do end up getting, the final electron acceptors, NADP+, and it gets reduced. It accepts that electron and gets reduced to NADPH. So that's good. So the green sulfur, they're like, yes, we can get NADPH being made. That's good. Unfortunately, the artist, again, I take issue, they keep forgetting. It's like, no, put the ATPase in here too to link this together. So I put it in words here that these bacteria also, through this process, there is a proton gradient that gets established, which does allow for ATP synthesis. So again, I think the artist kind of missed the boat here. They should have included that. They kind of forgot to finish the thought here that there is a proton gradient and you do get ATP synthesis. So even though it's one photosystem, they can get it to happen. And the added advantage to a lot of these bacteria is this wonderful chlorosome structure that is so good at absorbing light energy. So here on the left, they're just showing, again, they're trying to show that when, and it's not a chlorophyll pigment really that's in the reaction center. They label it here BCHL. The B stands for bacteriochlorophyll. All right, so it's a bacteriochlorophyll pigment. It's very chemically similar to chlorophyll, but there's some minor tweaks when you look at the actual chemical structure. But the end result is this bacteria chlorophyll's in the reaction center. It gets light energy hitting it, and then it excites one of its electrons off and goes through this very short electron transport chain process, ultimately to NADP+, and you get your NADPH. So even though they only have one photosystem, they can still get their needs met. They're able to get NADPH and ATP, which is necessary for them when they start to make their sugars. All right, so that is one group of organisms where they use photosystem 1. Then there's another group of organisms called the purple bacteria, and they only use photosystem 2. All right, so they don't, again, they're just down to one photosystem. And so, you know, remember too, microbes evolved before plants. And so these versions of photosynthesis occurred first, and then plants and cyanobacteria evolved a little bit more sophisticated processes in order to run their light reactions of photosynthesis. So purple bacteria have just photosystem 2. All right, now they're at a little bit of a disadvantage when you look at this carefully. So here's photosystem 2, and you can see it's getting hit with light energy. You see again, it's a bacteria chlorophyll pigment that gets that light energy. The electrons in that pigment get excited and boosted off to an acceptor, and then there is this weird process where instead of what we just looked at, we had a linear flow of electrons from the pigment to NADP+. Okay, so we call that linear. It's kind of like a one-way passage of electrons from start to finish. However, when you look here, you'll see the electron flowing to here, and then you see it coming back. So it is reducing, there is electrons, enough electrons flowing through here where NADP+ or NAD+, one or the other, will get reduced. But we call this a circular flow of electrons. And you also see when you look here, you're like, okay, where is, I don't see like hydrogen sulfide getting stripped of electrons anywhere. All I see is the electron in the pigment getting excited, and then you follow the arrows around and the electron comes back to the pigment. So what they're saying here is there isn't an ultimate electron donor. There's certainly not water being stripped of electrons because it's anoxygenic. You don't see any hydrogen sulfide or any other organic molecule here where they're showing it's donating electrons into the system. So it's really quite interesting. The electron in the pigment gets boosted off, but then it comes back and fills the gap, so to speak, gets hit with light energy again, goes around, and zings around, and this happens over and over. There's many of these complexes embedded in the microbial membrane. So you do end up getting NADH or NADPH. Well, not directly made, but it does get made. Okay, so I guess really when you look at how photosystem 2 is doing this, one of the big payoffs is that as the electrons are zinging around, getting excited, going through this electron transport chain process, but it's circular. So it comes back to the pigment, gets excited again, boosts around, zings around. You can imagine this happening over and over. And you can see these protons are getting pumped across the membrane. So as this is going around and around and around, you get pumping of protons, pumping of protons. So you get a nice proton gradient, which again, as you know, will then allow for ATP. So that's a huge payoff for this way of doing photosynthesis. But when you look carefully here, especially at this lower diagram, you're like, well, where's NADPH here? I don't see it. They just have this little box saying, oh yeah, NADPH will get made, but it's not directly made through this process. You get direct ability to make ATP through this process in the sense that you get a nice proton gradient, but they're saying you have to use reverse electron flow in order to get your NADH or your NADPH. And again, I'm not going to go into details on that. I talk a little bit more about it in Gen Micro, but basically one of the downsides of being a purple bacteria is you cannot directly get NADPH made in the light reactions. You actually have to expend a little bit of energy to get your NADPH. And so they are using light energy for that purpose. It's just not as efficient as what we were just seeing with photosystem 1 type bacteria or how plants do it. So again, I'm not going into detail, but if you can understand that, okay, purple bacteria, they're kind of a little bit of a disadvantage because they can't directly make NADH through this process. It's not that efficient. Certainly they can get ATP, but not the NADH.
So would purple bacteria be a little bit more acidic internally than most other organisms?
Acidic,
Just because they have so many protons floating around, it seems.
well, they usually the protons immediately get used for for ATP. They need a lot of ATP, and so that proton gradient. Well, okay, I see what you're saying. That I don't know. That's a good question. I don't know. I'm sure somebody has looked at their internal pH by now, but I don't know if it's dramatically different. I have not. I don't remember reading anything about that or learning anything about that in my years of study on this. Mostly when you learn about the purple bacteria, they're really interesting in the sense that, it's a very early process of photosynthesis. It's probably one of the first photosynthetic organisms. And so you see that, they're kind of limited in what they can do. And the downside of being a purple bacteria is you aren't terribly efficient at making NADH or NADPH. You can do it, but you have to expend a little bit of energy. So let me see if I have, let me just check. Whoops, come on. Where's my, oh, no. So when you're studying this, if you have to go back to this recording and just, kind of what we were just discussing here, how I was comparing and contrasting and pointing out key parts to each of these different ways of doing photosynthesis. Focus on, what's similar, what's different. I tend to like questions that I ask you on quizzes or exams where I might say, compare and contrast these two different ways of doing something. And so as you're studying this material, I think it would be really a good idea to approach it that way, where you're, kind of looking at all three, and doing this, okay, what's similar, what's different, and being able to express that. Ultimately, you're going to have to do that, via typing it out on a question or an answer on an exam. Any other questions on the different flavors of photosynthesis?
So do purple bacteria not have a final electron acceptor?
No, not really, because they have, that's another key thing. They do circular flow, whereas, photosystem 1, it's we call a linear flow. So the green bacteria have like a starting point where you have hydrogen sulfide, in this case. That's your ultimate starting point of where the electron starts from. And then the electrons get stripped off of that and get fed in. And then they end up at the end at NADP+. And so we say that's linear from, hydrogen sulfide all the way over to NADP+. And you don't see the electrons circling back in any way. But with photosystem 2, it's this circular flow of electrons. So that's a huge difference compared to all the other ones we just looked at. All the other ones are linear. And so that it's really quite different. And then of course they just aren't quite good enough to directly make NADH in this process. The energetics of it, when you actually look at the thermodynamics of it, it's just not quite good enough And so they have to push it using a little bit of ATP and expend some energy to get it to happen.
All right,
But
thank you.
Yep, the goal is still, you know, they got to get some ATP, they got to get some NADH or NADPH so that they can use that to make sugars. So right now we're just focusing like on part 1 of photosynthesis, where they have to get the necessary molecules that they can feed into their pathways to make sugars. So I'm not even going to really get into the Kelvin cycle and making sugars. I'm more interested that you understand the light reaction idea in looking at similarities and differences in the light reactions. Now these next slides are a lot of words. So these are posted, you know, don't rewrite everything that's posted, but I do need to show you these because when you're in a microbiology world, people will throw these terms at you. And so it's good to get comfortable with them. So the way I've typed this up, I've grouped them in pairs. So here's the first pair. So when we look at microbes as a microbiologist, we say, if we're trying to get to know our microbe, like, oh, this looks like a new microbe. I've never seen this one before. I wonder what it's all about. What we care about is, okay, what does it use for its carbon source? Does it use CO2 or does it use some organic molecule? How is it getting its energy? How is it getting its electron? So I'm grouping them here. So the first two to compare would be an autotroph versus a heterotroph. So again, this compare and contrast. If you're an autotroph, you are lucky enough to be able to take in CO2 and make your sugars like plants do and cyanobacteria and so on. Great. If you're a heterotroph like us, we're not so lucky. We have to eat something that was once alive in order to get our carbon source. All right, so it's either you're an autotroph or a heterotroph when it comes to how do you get your carbon. All right, so that's how you answer that carbon question. Let's look at the next idea. So then if you're looking at, well, what kind of energy are you using? If you're trying to get to know your microbe, as far as we know, the only two options are phototrophs or chemotrophs. So plants, cyanobacteria, you know, purple and green bacteria, they're phototrophs. They use light for their energy source. We aren't so lucky. We have to use chemicals. So chemotroph means chemical using. That means we have to take some kind of chemical compound and strip electrons off of that and use that as our energy source. Then you say, okay, well, where specifically are they getting their electron sources? And so your options, as far as we know, are you can either be a lithotroph or you can be an organotroph. And so litho is rock, troph to eat. So you're a rock eater if you can strip electrons off of inorganic molecules like hydrogen sulfide or hydrogen gas. We are organotrophs, so we have to use organic molecules in order to strip electrons off of them. So I think it works so much better if you can memorize these terms in pairs like this and understand what, you know, so the first two bullets are looking at your carbon, the middle two bullets are what's your energy source, and then the bottom two bullets are what specifically are your electron sources. And I think if you memorize them this way, it makes a whole lot more sense. I wish I would have learned this way back. I didn't get taught that, and it was confusing. These words are big and confusing. But if you can group them this way in your head, I think they make a whole lot more sense. So this is kind of like the foundation of these terms. But then, because microbes are so amazing, you have categories then where you can take those words we were just looking at and you can start to combine them a little bit and think about how are they doing this. So, for example, we know there's microbes that are photolithoautotrophs. So just dissect the word here. Photo, well, that's referring to light energy. Litho, well, that's rocks. So they must be using inorganic for their electron source. And then autotroph means they can make their own sugars. So that means they must be taking in CO2. So again, if you learn the previous slide in pairs, and then as you see these more complicated terms, you can start picking the word apart and figure it out. You don't even really have to memorize it. So the photolithoautotrophs in the microbial world, that's the cyanobacteria and the purple and green bacteria we were just looking at. They are definitely photolithoautotrophs. Chemoorganoheterotrophs, so chemicals, they're using chemicals instead of light for their energy source.
I have a question real quick.
Yes.
would the purple bacteria be lithotrophs if they don't actually have an inorganic or like an electron source?
Oh, that's a really good question. And yeah, it's like, okay, how strict of a definition are you getting with the purple? They lump them into the photolithoautotrophs. Purple are kind of in their own little category though, really. I mean, because it's just the pigment electron that's getting excited, but the electron comes back and gets re-excited. And so it's, yeah, I get your point. It's kind of a oddball. It's kind of like an outlier, right? It's kind of a photolithoautotroph, but not exactly. So you can think of it that way, and I think that's accurate. It looks like we're running out of time. Let me just finish the thought here. Chemoorganoheterotrophs, we'll stop here. They use organic compounds for their energy, electron, and carbon source. So we are chemoorganoheterotrophs as well. That's why a lot of pathogens that attack us, they're also chemoorganoheterotrophs. So they're well adapted to our type of body because they do their pathways very similar to how we do ours. And so that makes them better at attacking us. So anyways, so we'll stop there. Just a couple more terms. And I didn't even get into the next topic. We're pretty behind schedule. I'm going to have to shorten up some of this future slides because I want you to get some content before you start doing your independent research projects. You have to get some key knowledge before I cut you loose on that. So remember, you still have the field guide issues coming. I hope you're making progress on that. Ask me if you have questions on it. And then we have to start talking about getting an outline and getting an idea for your independent research project. So I can talk to some of you tomorrow in lab. So group 1 is coming into lab tomorrow. All right, I'll see some of you tomorrow. Otherwise, be careful and have a good weekend for everybody.
Yeah, that first thing that you mentioned, not like the big project, the independent research, but the other one,
The field guide.
what was
Yeah.
that again? So I have an idea of where I want to go. It's a pretty sweet spot, but so what do I do as of right now? I have that in my mind. Do I have to like go there and get some samples right now?
You don't have to sample. For the field guide, I am not going to ask you to sample. You can, if you want to use that field plot for your actual independent research project later, you can, but for the actual field guide assignment, I will not expect you to take any samples. However, yes, you have to go out there, maybe more than once, because you're going to have to go out there and assess your site visually. And, you could take some measurements like what's the temperature of the water. You could estimate, how are there really shaded areas versus areas that seem to get a lot of consistent sunlight. Well, that will impact who lives where in terms of the microbes. So really assessing your site visually, you could take some brief measurements if you want, but not actually will I ask you to like go take a soil sample and figure out what microbes are there. I'm not going to ask you to do that.
Okay. And I guess I've been studying for this class, but I haven't I gotta read the rest of those readings. And then, so where's the link to this this kind of exercise right here?
The field guide?
Yeah.
The syllabus says a few things. If you scroll down, I think the last page of the syllabus, I talk a little bit about it. And then I'll see if I have another handout that I can post into EduCat. I might have something with a little bit more detail. But so read what it says on the syllabus, and then we can talk some more, or you can email me with some specific questions that you have on it, okay?
Okay, so we should be working on that right now though, right? Like the field guide?
Yeah, because it's due in October.
Yeah.
And it's already September.
October 16th.
Yeah, and it's, October or September 10th right now. So, we all have busy lives. I'm trying to prod you to remember that you got to get that done, which means you have to go to find a site, and then you'll probably have to go to it a couple times, I would imagine. So
what is like the specific microbes? Okay. So am I just typing like a formal report about this or something?
No, the field guide is more, I'm asking you to write it not like a lab report. I don't want that. I want you to write it to an audience. You have to pretend you're writing it to people. Let's say there's people who come up in the summer or fall, and they're like, I want to go hike somewhere in Marquette, and they go to the Chamber of Commerce, and they would have your field guide there, and they'd say, oh, you want to go visit a cool spot and you like microbiology? Well, here, check this out. And so they would give them your field guide, and they would take that, and on that field guide, they would have directions on how to get there. And then once they get there, through your field guide, what you type up and the pictures that you provide, you would describe that site. You would say interesting things about the microbes. So I actually want you to almost approach it like you're marketing or advertising and trying to excite people about why we should care about microbes at this site and what might the microbes be doing at this site. And so that's more, so it's a little different than what you're used to in a science class. And I know that some students are a little freaked out by that, but truly, I don't want a lab report. I want you to use colorful adjectives and try to find a way to write where you're trying to excite the person. Oh, this is cool. I learned so much. This is great. So you're writing to an audience of people who are avid outdoors people who have some knowledge of biology, and in particular, they're interested in microbiology and what's going on in microbial ecology.
Okay.
Okay.
Gotcha. All right. So it's more of just like have fun with it, but then actually do a really good job. Like,
yeah, it has to be accurate. In fact, certainly you have to, and you have to cite the literature. So it's really an exercise to get you into the published literature, because you don't know all this stuff off the top of your head. You're going to have to go into this literature and look for papers that are published on stuff similar to the site you're looking at. Okay, like what's happening in this pond? What might be happening in this pond? Well, I'm going to go into the literature and find out what kind of aquatic microbes are in lake systems or pond systems. Oh, I see all these lichens on the tree. Well, what are lichens again? So you'd have to look into that a little bit. When I put a stick down into the stream, boy, I smell rotten eggs. Well, what's up with that? So sometimes you can play around with your site a little bit, like sticking especially sticks or paddles into a lake bottom, and you get the silt stirred up and you smell some interesting gases. Well, there's plenty of microbes there that you could then do a Google search, in the papers, and you could cite what might be there. So I would like everyone to go to their site at least once and then come back and start asking me questions. I can prompt you to look in certain areas to get more information about it.
Okay, sweet. Thanks for the advice right there.
Yeah. Anything
All
else from anybody else on the field guide or?
So this isn't the field guide. This is more like the lithotropy and stuff. Would radiotrophy be on its own little class?
Yeah, I would say so. Yeah, that would definitely be its own little group.
And that counts as an electron donator. What would that be? Energy source? Because it's almost like instead of like phototrophy.
Yeah, I know what you mean. It's part of the electromagnetic spectrum. So would they call that? It's not technically photosynthesis, though. We have a very strict definition of what photosynthesis is. So yeah, it's kind of its own little grouping there. I'd have to, the most recent textbook that I have, I don't even know if they talk about those microbes in that textbook. It's probably
it's mostly fungus, and it's mostly melanin that does the conversions.
Yeah, I'd have to. I don't know if you have any source or information that you can point me to. I'd be interested. Because I don't have anything that I'm aware of. I don't even think the textbook that I use. I have a big, huge textbook on microbes. The microbiology textbook, and I don't think they mention anything about it.
Yeah, it is super specific. So.
Yeah.
Interesting though.
Oh yeah, microbes they're mind-blowing at what they can do, and then we're still discovering bizarre things that look at this, look what this microbe can do. I mean, it's just
like fungus are my thing, and like chanterelles alone, like there's like, as someone who forages them, like they have there's 3 different species, but they only classify them as one. But like if you actually go out and forage them, you're like, no, these are way different. Like they taste different, they look different, they smell different.
Yep. Uh-huh. Yeah, it's a never-ending. You cannot get bored if you study microbes. There's no way to get bored because there's just so much diversity and interesting things that they can do. It's, you know, it's almost like stepping into the world of science fiction. It's really incredible. I mean, every week, if I'm reading something, you know, current in the microbiology news world, there's always something new and bizarre. And so I think we'll just continue that. I don't think we'll ever run out of things that we say, wow, look at that.
And especially because they reproduce so fast that there's like a lot of evolutionary pressure and everything.
Yeah, exactly. Yeah, their ability to evolve is so much faster than what we can do. Absolutely. And that, you're right, that creates a lot of diversity and really unique adaptations. So, okay, I'm gonna.
Yeah.